Recording is started. Yeah, so thank you everybody for coming to this webinar. I'm Betsy, I'm an editor at the Sick Times, and I'm excited to be moderating this event talking about wastewater surveillance, kind of giving a bit of behind the scenes into where those data on the dashboards that so many of us love to look at and rely on for COVID information and other disease information, talking about where that's coming from. Yeah, so just to give a bit of intro, go to the next slide, Ellie. Yeah, so I mentioned I'm Betsy. I'm a co-founder and editor at the Sick Times, which is a nonprofit news site focused on long COVID, COVID-19, and related chronic diseases. So we use wastewater data a lot in our reporting. I write a weekly COVID update that goes in our newsletter and on our website that is very, very reliant on this information in terms of tracking both COVID and other diseases like the flu and RSV and other emerging threats like Mpox that we might talk about a bit today as well. And so it's, a very valuable source, but also a very complicated one and one that scientists are still learning a lot about. So I think we'll really get into all of that today with Dr. Alexandria Boehm, who is our other speaker. Yes. So just to go over some ground rules, guidelines, we're taking the National Association of Science Writers Conference and Meeting Code of Conduct as a model here because they do a lot of online events like this. So I really wanted to just emphasize, staying respectful, considerate for everybody who is attending and speakers of the event, being thoughtful and respectful and discussing or debating ideas. And just keep in mind, if somebody is like spamming comments or if something is inappropriate, I or one of the Sick Times or WastewaterSCAN folks helping out behind the scenes here, might ask somebody to stop a behavior. So just, please be respectful if that happens. And then some logistical notes. So we are recording this, as you might have noticed. We're going to be sharing the video on YouTube after the event. If you need captions, you should be able to enable that in the Zoom settings. Let us know if you need help with that. And then you can put your questions in the Q&A box. So please don't use the chat. The Q&A function just helps us to keep track of questions as they're coming in. And folks can also upvote each other's questions if there's something that you really want us to answer. And if we can't get through all the burning questions during this time, we'll try to answer questions later with a written Q&A. With that, I'll turn it over to Allie to give more of an intro to WastewaterSCAN.
Hi, everybody. Thank you for joining today or watching this video at a later time. I'm going to give a brief introduction to the WastewaterSCAN data, and then I'm here to answer questions mostly. WastewaterSCAN is a national effort, and it's based at Stanford University. And we've been building a national network for doing wastewater surveillance since the beginning of the pandemic. And we use consistent methods across all of our wastewater treatment plant partners that we work with and provide that data to public health to inform their responses locally and nationally to different diseases. So I'm the PI of the project. I'm Allie Boehm. I'm a professor at Stanford University. Marlene Wolf and I are both program directors for WastewaterSCAN. We contract to Verily Life Sciences to run the laboratory analysis for this project. That was a partnership that began in 2020. At that time, Verily did not do any wastewater measurements, and we worked with them to sort of train them on the approaches that we wanted to take for monitoring wastewater and worked with them to teach them how we wanted the measurements made, and they've grown into an amazing partner for this project. We also work with the National League of Cities for capacity building. And of course, we're very reliant on all of our partners, the municipal wastewater treatment plants, as well as state and local and federal public health officials. A quick overview of the program. We work with 148 plants across the United States, and those plants serve about 11.85, not about, exactly 11.85% of the US population, and we work with 40 states. The states that we don't work with most of them have their own wastewater monitoring programs as well, but they're just not part of our project. The pathogens that we currently test for are listed here. We test for a variety of respiratory pathogens, including SARS-CoV-2, of course, influenza A, B, as well as the subtypes H1, H3, and H5, which is the avian influenza, the H5, RSV, metapneumovirus, and enterovirus D68. We also measure 2 enteric pathogens, norovirus G2 and rotavirus. And then we also measure Candida auris, which is a fungal pathogen, hepatitis A, and Mpox clade 2. And so this is not the clade of Mpox that is currently causing a lot of news, media, and concern that's circulating in Africa. That is clade 1. So we do not measure that at this time. And we also sequence SARS-CoV-2 RNA from about 30 sites. So we're able to say something about the different variants that are circulating. And then we also run some controls, PMMOV, which is a pepper virus that is found in a lot of our feces if we consume any kind of pepper products. And then we use a bovine coronavirus as a control in our laboratory methods. I just wanted to show you this quick timeline that describes how we started and where we're at now. So we started in March 2020 when we started doing research at Stanford University, detecting SARS-CoV-2 in wastewater solids. And then we scaled that effort across the Bay Area of California, expanded the types of things we were measuring to different variants, to influenza A and RSV. And then we started expanding nationally across the country in 2022. We started measuring Mpox as well as other pathogens, doing sequencing, and then work with communicating those results by providing the categorizations that are on our website, which I'll talk about. And then most recently, we've added monitoring for different influenza subtypes, including the avian influenza subtype, the H5. All of our work is grounded in the scientific process because we are based at a university. We publish all of our work. It goes through rigorous peer review by other scientists. And I know you're not going to be able to scan all these QR codes, but we have peer-reviewed papers on all these topics, which you can look at if you'd like. They're all open access. And importantly, Amanda Bidwell, who works with us on this project at Stanford, she has put a lot of effort into summarizing all of our peer-reviewed papers into a more layperson type description of what we do. And so that is accessible at the website listed here, which I think she has put in the chat, as you can see, if you're interested in poking around and reading a little bit more about the science. So I know there's a lot of questions on the categories and how does WastewaterSCAN determine different categories for wastewater. And so I'm just going to go through that very briefly before we turn to like more specific questions that you all have. But what we decided when we started the categorization process was that we wanted to use information on the level of the viral nucleic acids in wastewater and the trend. So if it's going up, flat, or coming down, as well as frequency of occurrence of positive detections to describe the wastewater category as a low, medium, or high. So that is what we aim to do with this. And so, for example, I think intuitively, if you look at this plot here, which shows SARS-CoV-2 over time, you can see easily that things are going up here. That's kind of not ideal. Don't want to say bad, but those of us concerned about getting COVID might be concerned when it's coming down. That's kind of good, like we're feeling a little better. And then when we're at very high levels, relatively high levels, we might feel more bad about it. And so that's sort of what we wanted to capture in a more scientific, rigorous method. And so we are using what's called tertiles. So we ask, is the level in the lower third, the middle third, or the upper third of all the measurements we've ever made in order to categorize it as a level, for example. So we have 3 different categories of pathogens that we monitor that get slightly different logic on whether it's categorized as low, medium, or high. The first are pathogens that are commonly detected. They're always in wastewater, regardless of the season, and that is SARS-CoV-2, norovirus, and rotavirus. And those get their categorization as low, medium, high in a certain way. And then we have seasonal pathogens that are usually not circulating except during, in this case, more of the winter or springtime. And for those pathogens, they get a different way of being categorized as low, medium, or high. And then the third category are pathogens that are not commonly detected. This includes Mpox, clade 2, EBD 68, Candida auris, and hepatitis A. And they get categorized as low, medium, and high based on a different logic. And I'm going to go through that logic right now. So for the pathogens that are commonly detected, what we have established is this categorization system. So if the level is low, in the lower third of what it's been over all time across all the plants, and the trend over the last 21 days is downward or no trend, it goes into a low category. And then we have the medium category when we have relatively low levels or middle-ish levels, regardless of the trend, get into the medium category. And then the high category is when we have relatively high levels, so everything in the upper third, regardless of trend, and if it's in the middle third and upward, that gets a high category. Now, this is somewhat subjective. The wastewater level and the trend are quantitative and scientific, so there's no question when you look at the data and you can say the level is this category here, one of these categories, and the trend is one of these categories. What is a bit subjective is then how we assign it to a category. So for example, somebody might say, well, if it's in the lower third, even if it has an upward trend, I would categorize that as low. Somebody might say that, but we chose to categorize it as medium. We did share this with some of our public health colleagues and get their input and recommendations. And this is what we landed with for SARS-CoV-2, norovirus, and rotavirus. So you can see an example of this categorization on our website. This is the national overview across the country of the category for SARS-CoV-2. And you can see an aggregate across the United States that we're in the high category. We have a downward trend in the last 21 days, but we still have a high concentration. So that's why we're still in the high category for SARS-CoV-2. But you can see there are some locations where we have a blue. So that means at those specific locations, we might have a low category for SARS-CoV-2, but taken all together in aggregate, we're in a high category across the entire country. And you can access the number of sites in each category. It pops up when you hit that little I on the info on the website. Now, there is this not calculated category, and you can see that some sites are gray, and that means that the plant may not have sent in a sample recently. They may have like a staff member who's ill, so they aren't able to get the sample. And so if there's no data within the last 7 days, the category is not calculated for that site. For the seasonal pathogens that are not commonly detected during most of the year, they get a low category unless they're in seasonal onset. And I'm not going to go through all this logic of the seasonal onset, but basically, if the concentration is non-detect most of the time, then we just consider that in seasonal offset and it will always be low. And then once it hits onset, which depending on the virus might be in like November for influenza, then it starts getting categorized just like SARS-CoV-2. So looking at the trend and the concentration. And then for the pathogens that are not commonly detected regardless of season, we just look at the frequency of occurrence. So if we have less than 50% positive detections in the last 10 days, we say it's low. And then we have this other logic. So if all samples are positive in the last 10 days, for example, it would be high. So these are for the rarer pathogens like Mpox, Candida auris, Hep A, and EVD68. So that's it for the presentation. And now we'll get to the questions, which is the reason we're really here.
Yeah, thank you for that intro. I think it's so helpful to see all of that laid out, especially the the different approaches between SARS-CoV-2 and the other pathogens in interpreting the data. I guess building off of that a bit, I wanted to start with a question about, how you approach interpreting this data, and also for people who might be watching and want to talk about wastewater surveillance or want to talk about the results with their friends and their communities. Like, how would you suggest people approach this? In terms of, like, sometimes it can feel a little abstract, right? So what strategies do you have in talking about it?
Yeah, that's a good question. I think I find that talking about the categories of low, medium, and high is pretty, people can relate to it and understand, um, what that means. And I think then the question is, well, what does high mean and what does low mean and what does medium mean? And I think, we have to be a little careful. I remember initially going through this and talking with people about like the terror level. If you guys remember that from years ago when there was a terror alert and we were always in like orange and it loses its meaning, right? If you're always in the orange category. The categories aren't necessarily a risk category. It's just sort of saying, over in my mind, it's like over the period of time that I can remember, which is maybe a year or two, that COVID is circulating at sort of a higher level, and we should be concerned about potentially transmission during periods of high and medium, whereas in the low levels, like there's reduced potential for transmission. So that's how I think about it. But we do want to be careful because, like, I'm a scientist, and we want to be really careful about the subjective interpretation of wastewater. I know that doesn't help people, but I think, if you're risk averse and you are planning like a vacation or visiting family that are immunocompromised, for example, you want to be extra cautious during the high period. Wear a mask on the train, on the plane, at the grocery store, and just be really cautious because it means that there's a high likelihood that somebody in the store with you or on the plane has COVID, and you don't want to get it because the consequences to you are high, maybe. When you're in the medium category, depending on your risk aversion, you might be more comfortable not masking. For example, and then the low category, depending on your situation, you may not be interested in masking at all, even on a crowded airport, for example. So those are some of the things that I try to talk to people about when I explain the categorizations.
Makes sense. Yeah. So another thing I wanted to ask about in terms of analyzing, interpreting the data is all of the factors that can impact those numbers that we get from wastewater. So I often try to remind people that it's a population level metric and we're measuring, you know, uh, material from the environment. So it's not just like human waste that's in there. There's a bunch of other stuff that goes into our sewer systems. And also our sewer, like those systems are very different from place to place and all of this. So what are some of the most important factors that you think about or that you would say are important for people to know about if they're looking at these data?
Yeah, another great question. I think when we started this work, we expected that site-specific differences and uniqueness of the different wastewater treatment plants would give rise to just really variable results that could not be compared across location or aggregated or anything. And to our shock and surprise, it seemed that, we could and we could aggregate things, we could compare things that, if we normalized our measurements by PMMOV, which is something that we do, that everything would sort of fall on the same line in an incidence rate versus COVID and wastewater plot. So it's really surprising, to see that. So I surprisingly, like, I can't tell you, um, something that really stands out. We thought that wastewater treatment plants that were combined sewer overflow systems that had stormwater going into them would behave very differently from other plants, but we don't have a lot of those plants in WastewaterSCAN. We haven't seen a huge difference. We thought that sampling from the primary clarifier versus the influent would be a big difference. It hasn't been. I would say like the biggest challenge in socializing this data is the fact, as you already said, Betsy, that these are environmental data and they vary in stochastically and deterministically in ways that people who are used to looking at like positivity rate data aren't used to. So if you're looking at the data and you see this really high data point, just sort of an outlier, you know, it doesn't mean that's wrong or it's an error. It's just that the environment is like very heterogeneous, and so is our feces. And so you're going to have this variability that is inherent in the data. So that's like the best thing to, I think, keep in mind, is not to discount the data because of the variability, and just understand that is inherent to an environmental data stream. And those measurements that seem like outliers are just as important as the measurements that don't seem like outliers.
Yeah. I mean, that point about how the data varies, I think also brings me to another question, which I know a lot of people had both in the registration form when folks were submitting questions, and I saw in the Q&A pop up too, is this potential process of like translating wastewater results to estimated infections. There, you know, for folks who are in like COVID cautious spaces on social media, or sometimes articles will cite these kinds of estimates. I'm curious, like, what you think of these, or if this is something WastewaterSCAN has worked on at all. My understanding from reporting is that it's pretty complicated and hard to do, but you're the expert, so I wanted to ask you about this.
I think that when COVID-19 testing was really commonplace for people who were experiencing severe symptoms and test seeking behavior was great and high, that empirical relationships between the wastewater and the incidence rate data was really predictable. But since testing has dropped off so much, it's, I guess, we can use those old relationships to estimate how many people are presenting with severe illness. The caveat to that, though, is that different variants might cause different severities of illness. So I think, you know, probably a lot of people in COVID cautious spaces are familiar with with some concepts of epidemiology, but you know, SARS-CoV-2 can cause infections and people can be asymptomatic. They can be mildly symptomatic, or they can be severely symptomatic, or, you know, hospitalization, et cetera. So all these different categories of infections. And we never knew for sure how many people were infected across all those different categories. We only had data really on people who were moderately to severely ill with COVID who sought out testing. So that's why we were able to see nice correlations. So now I think the panacea of wastewater surveillance would be to come up with those estimates of how many infections there are from the wastewater data. And there's a lot of information that is almost variant specific that we might need to know, like the shedding rate from different populations. From, is it age dependent? Is it dependent on the severity of symptoms? So there's those complexities related to like the human body. And then there's also complexities associated with the waste stream. Is there within sewer decay of the RNA? We've done experiments that suggest there's not. But, you know, in some situations there could be. So there's sort of the environmental modeling aspect, but also the human like shedding aspect. And that data seems to come out much later than when we want it, because you have to enroll subjects, and that work takes a long time to do in the medical sciences.
Yeah, yeah, that makes sense. Yeah, you mentioned the viral shedding, which is another question we got in terms of like how variants can impact this. what do we know, or what do we still need to investigate there?
Yeah, I have seen some recent work, really nice work, come out on the differences in shedding profiles across different variants. And so there are statistically significant differences in how much RNA is shed with different variants. But my assessment is that the effect size was small. So there were differences, but they weren't huge. It wasn't like an order of magnitude difference. So,
Mm.
um, based on the data and how it continues to show us, like, trends and levels, and how that seems to agree with, like, what doctors are seeing and hospitalizations and that kind of thing, I don't think it's enough to, like, throw off our wastewater measurements and make them not comparable across time. But it will be really interesting to see how that all shakes out. And we could go back and, I'm a nerdy scientist, so I want to go back and, like, see how the predominant variant might have affected that. But that more data on shedding across different variants, I think, would be really helpful.
Yeah. What would those experiments look like? Like, I'm curious what
Yeah.
it
So
seems like hard to measure.
Yeah, so this is how you would do the experiment. So first you would have to obtain funding, probably from NIH or CDC, and then you would have to get an IRB ethical approval, and then you could start enrolling patients. So you would, like, on social media or whatever way the IRB, the ethics committee, says you can, you would recruit patients. So you would say, do you have COVID-19, or does someone in your household have COVID-19? Email us and enroll in this study, and we'll come by and pick up a fecal sample, a sputum sample, a urine sample from you every day, and we'll drop off these little kits. And so if you're enrolled in the study, you might get a gift card from Amazon, and then every day you leave a gift for the researchers. So you would have to take your specimen collection vial into the bathroom. When you poop, put some in the vial, pee in the vial, spit in the vial, put it in your freezer, maybe wait for them to come by and pick it up. The researchers would come by and pick it up when they got all their, like, 200 participants from the study. They would then have to analyze all the samples, work up the data, write a publication, and submit it. So it requires, human subjects to be enrolled in the study, and humans are complicated things. So getting them to comply with the sampling and everything can be tricky. But that's how the study would work, most likely.
Makes sense. I
Mhm.
also wanted to note, as we're talking about risk levels and how to interpret data, just a comment that came up from the Q&A, which is that, you know, everybody is susceptible to developing long COVID, even after a mild
Mhm.
or asymptomatic infection, and it can happen for people who have pre-existing conditions or health risks, or for those who don't necessarily have anything that they know about before COVID. So when we're talking about these levels, it really is relevant to everybody, although certainly certain segments of the population, like folks in our audience at the sick times, are maybe more hyper aware of this than others. So
Yeah, and I think some people just don't know, they
yeah,
don't understand. So um, I agree with that.
yeah. Yeah, I mean, we published an article recently. This is a bit off topic, but an article talking about the relationship between hypermobility and long COVID, and sort of pointing out that this might be like a genetic susceptibility that people might not know they have until they're kind of dealing with symptoms later. So there's
Mhm.
a lot to be cognizant of. Um, yeah. So another kind of process question that I think came up in the responses on the registration form is, like, what happens in between the sample being collected at the wastewater treatment plant and then that, like, the data point making it onto the dashboard, and how that might contribute to, for example, the length of time between the sample being collected and the dashboard being updated, and also factors of, like, the data analysis that go into that.
Mhm. So this last part of the question is really easy. The data analysis is all instantaneous. It's done on GitHub, and as soon we have a pipeline where as soon as the data are available in the lab, they get uploaded to the website. All the calculations occur instantaneously. So that we've worked with the web developer to do, and none of that is involves a human, luckily. And
Very efficient.
Yeah. So the part about where we involve humans is where things can get a little tricky. So we do rely on a human to get the sample at the wastewater treatment plant, package it for like UPS or FedEx to pick up, and then they will pick it up and deliver it to the contracted lab. That's overnight, so that takes a day. It is possible that someone collects the sample and doesn't call the UPS carrier until like the second or third day, and it sits in the 4-degree C room. Um, but most of the samples are processed by the lab within 48 hours of sample collection. And then it takes, maybe 8 to 12 hours to process the sample in the lab or less, and then it gets uploaded to the site right away. There are a few sites where that sample processing is not 48 hours, but 24 hours, where the sites are like within really close driving distance of the contracted lab. So the data are flowing to the website daily, across all the participating plants. So that's how that works.
I see.
Yeah.
And so when the data flow to the website, my understanding is it goes first to the local trends for the specific sewer sheds, but then there's also the process of getting the regional and national averages.
Mm-hmm.
So what is that process like? And also maybe a related question that we're getting in preparing for this webinar is like why you sometimes might see different patterns or different levels on WastewaterSCAN dashboard compared to other data sources like the CDC or BioBot, I think of as the kind of main ones that everybody looks at.
Mm-hmm.
And so like as you mentioned these pieces about timing and like which sites are represented, all kind of plays a role in those eventual trends we see. So I was wondering if you could talk about that a little bit.
Yeah, so the aggregation across sites also occurs instantaneously. And so, we only calculate an aggregated result when a certain fraction of the data and that aggregation are available. So like if we're aggregating across the country, this is like a really obtuse example. And for some reason, only one site has data for yesterday. We're not going to produce a category using that one data point from yesterday. So we make sure that I think it's 50%, Amanda, of the sites have within the aggregation have some data to produce a new category on a particular day. So all of that is instantaneous, and the logic is all built into the website processing on GitHub. And we have it all documented on our website if people want to look at the exact logic of the coding for that. I think the really important question you asked is like, why do the data trends or levels sometimes look different across these different locations? And as you already mentioned, the representation of sites could be different in different aggregations. But what I think is really the most important thing is that our website updates daily, hourly, as soon as, constantly. And we pay for that at GitHub to have like the engines constantly running. Other sites, due to, availability of staff and person power, they may have a note that they only update on Monday, Wednesday, Fridays, or they might only update on Friday. So there's also, there could be delays. So, if we submit our data to the national database and they want to do some QA QC on it that involves a person, that might create a day of delay before that data ends up on another site. So that's all I can say because I'm not privy to like all the other sites and how they actually aggregate and interpret their data.
Yeah, that
And
makes
it's not
sense.
always transparent on their websites either.
Yes. Yeah, I mean, I know just to give the CDC as an example, because that's another national dashboard that a lot of people look at. They're aggregating from a ton of sites, but they're sites that all have different, or many of them have different approaches to testing and analyzing and processing their results. So the CDC had to kind of come up with a new metric to compare across those different locations, which is a bit different from the process with WastewaterSCAN, where you have sites that are all using the same process and the data is all going through the same pipeline. So there are a lot of layers of complication here, I think.
Mhm.
Yeah.
But I think importantly, like overall, I think they normally agree with each other once
Yeah.
they all catch up. So,
Yes.
if you
Yeah.
look retrospectively, like at last winter, and you compared everything, it would all go up and come down around the same time. And so it's just like that leading edge where people are really looking,
Yeah.
I'm going to Tahiti tomorrow. Should I wear a mask on the plane? That you're going to see differences because the site one site might not have been updated since last Monday, but this WastewaterSCAN site updated today or something like that.
Yeah. Yes, that's true. I mean, definitely as a journalist who is like writing updates based on the wastewater data once a week, I do notice that. But I
Mhm.
also see how it resolves kind of over time. Yeah, so also speaking of the resource challenge, another question that folks are asking with wastewater scan is, you all downsized a little bit in terms of the number of testing sites this summer. I believe it was about 40, 46 sites that are no longer being tested. Could you talk about, what happened there and, what the program is looking like going forward?
Yeah. So, I mean, our program is really focused on advancing the science of wastewater surveillance. I mean, we're based at a university. And so, what we wanted to do in this like next year for wastewater scan is really focus on some new science questions and translational issues. For example, like thinking about influenza, like we've really honed in now and we're measuring H5 and H1 and H3 for influenza. And we have to choose. There's so many dimensions to this project. There's the frequency of sampling, there's the number of sites, there's the number of assays. And, as scientists, we decided we wanted to like go a little bit more crazy on the number of things we're measuring. And the science really suggested to us that there was a lot of spatial coherence across sites that were very close together. So if a site in like the East Bay of San Francisco Bay went up, so did the measurements on the peninsula in San Francisco Bay. And for those of you not from San Francisco, like, yes, the geography is crazy, but that I live here, so that's a good example for me. But the spatial coherence means that, like, you don't, from a science perspective, the sites are very similar, so you kind of don't need to. And I think this is something that CDC is also working really hard with now is like, how do you right size wastewater surveillance? Like, do we really need a site? Does every wastewater treatment plant need to be monitored, or can we have sentinel sites that are representative?
Mm.
And I know, like, for individuals, they would like their own sewer shed monitored because they rely on that data. But the truth is, the sewer shed that's 100 miles away might give really similar results to what you would have expected at your local sewer shed. And so, in making our decisions about how to work over the next year, we use the science suggesting that spatial coherence was occurring, and also really wanting to focus on influenza and going a little bit deeper there on
Mhm.
the assays was something we decided to do for our research.
So it's kind of the research questions um, that
Yeah.
are yeah, I see.
Yeah, yeah. And
Yeah.
I mean, a really important part of this project is, which makes this a really unique project in academia, is we're not just doing science, but we're also very, very interested in translational work. So we really enjoy working with public health and CDC. So we still want to keep our connections with the public health folks, because understanding what they need to uh, sort of embrace wastewater surveillance and providing it to them and really working with them with this new data stream is something that we also want to do. So, we also considered, our public health partners and talk to all of them actually before we decided to not work with the site for the next year, to make sure that they agreed with us that that site was something that they could either pick up with their own state program, or that it was redundant from their perspective because it was so similar to the site right next to it. So
Mhm.
that was important to us as well.
Yeah. Could you say more about that like spatial cohesion piece? Because I think this is something that I also see people wonder about a lot, like asking, if there's not wastewater testing in my community or for my sewer shed, but maybe there is like a few towns over or for this big city, or if there, like, with people who are in situations where there's only one testing site like in their state, like how do you think about, or how should folks think about interpreting kind of what the data is available for them, and also like how that relates to the national and regional trends that we see, like to what extent are there potentially gaps in our knowledge there?
Yeah, I mean, that's like a really big question. I'll start with the first part, which is easier. I have students and postdocs who are working on this, and if you think about it from like sort of a disease transmission perspective, like things that are really contagious, where there's a lot of mixing of people, you're going to expect that sites that are next to each other um, are going to go up together because it just like if you have really contagious like influenza A and people are like moving around in this state, like everything's going to go up at the same time. And we do find that like even adjacent states, everything kind of goes up with influenza at the same time. um so so I mean, we are finding that for these common pathogens that we monitor, that there is like a lot of spatial coherence in the signal. Um, but there are some pathogens where that maybe isn't true, like Mpox, for example, might be where it's very rare, where you're really looking for that like first case or that first sign of an outbreak, where you do want a lot of different plants because you don't have a lot of spatial coherence. So I think this is like an ongoing research question related to the sort of frequency and transmissibility of different pathogens. Regarding your question about aggregation, national averages, and regional averages and state averages, um, I think it's tricky to know what we're missing until we have it all and can downsample and say, if we remove this, do we get the same result?
Mm.
We've done sort of a downsampling analysis with our temporal data to say, because there's some sites where we have samples every single day, and we've said, what happens if we didn't have that? What if we just had 3 samples per week? Would we get the same trend and stuff? So we've done the temporal downsampling, but we haven't gone back and done a sort of spatial downsampling. And so thank you for the great research idea, but I think that's something that we don't fully understand yet, but it's very relevant because even as I said, now CDC is thinking about how do we right size wastewater
Mm.
surveillance. And I think all the public, like the people on this call or who are watching this, will have their own opinions about like how important local monitoring is versus this idea of right sizing and downscaling.
Yeah, that makes sense. To that point also about collaborating with public health, I saw one of the questions in the Q&A is about sort of who are the intended users of the dashboard and of WastewaterSCAN's data, public like local health departments, members of the public. So how do you think about like the ideal use cases for the data, and also maybe what if you want to share like some of what you've learned by collaborating with public health officials in different places over the last few years.
Yeah. Um, so the data that we collect, we say we have like this data use statement on our website, and we do collect it. I mean, the goal of our project ultimately is to advance like the public good and maximize the public good of these data that we're collecting. And so that means that we're trying to show the data and interpret the data in the way that the public can understand, which is why we created the categories, and also make the data available to public health. And so like we have an API for our site, which is for public health to use. They will often show the data on their own dashboard. So there's a lot of data sharing going on with them. So we really, and we also think that our data are relevant for other academics who are interested in doing research. So we sort of embrace like a lot of different audiences here. But yeah, so it's hard to make something that's relevant to all audiences. I really think public health are making their own dashboards at this point, but early on, that was our main audience, and our dashboard was just for them. And then they eventually began to really value the data and make their own dashboards, which is like the biggest compliment when someone wants to show your data like on their website. That's just amazing for us. In terms of the last part of your question, which was, I can't remember exactly what it was, or did I answer
I
it
guess like
all?
lessons learned from public health. So you
Oh
kind of
yeah,
have answered it. Yeah.
yeah, it's been amazing working with public health and seeing the transition from them being completely, I will say, uninterested in wastewater data to being very, very interested in it over the last 3 to 4 years. It's been really great to be like right at that forefront of like science to policy translation.
Yeah.
Um,
Yeah, I mean, I think that's related to another question too, which we were kind of getting in the Zoom registration, but also I know has come up a lot in my reporting, which is like the long-term future for these programs, WastewaterSCAN and just like in general. Because I've talked to a lot of scientists like yourself who like started doing this in 2020, and now it's like become such a big part of our infrastructure for tracking COVID, and is starting to become that for tracking other diseases. But then there's also that desire for like public health departments to kind of take ownership of this. So I'm curious like how you've seen this evolve with WastewaterSCAN, and also what you see as the kind of continued future for the program, like that balance between the scientific questions and also the like providing of the public health service.
yeah. Um, it's a delicate balance, and I think like what keeps me and Marlene as scientists and professors interested in this space is that we're able to do new things and advance new ideas. Like our recent work with avian influenza was really exciting to detect that and have the detection sort of agree with cattle infections and all that. Um, so that's what keeps us engaged, is sort of like pushing the new science forward, which is why for the next year we're focusing on some new things that are completely novel uses of wastewater. um, I have been really happy to see how wastewater surveillance has been embraced by states and the CDC, and it seems to be something that is going to continue into the future. I can't speak to like the ongoing availability of state and federal funding for that, but I hope that it will continue. I think we've shown that it can be really useful. Um, and then WastewaterSCAN is like an academic project. So, we, you know, this is sort of an ongoing program, and we'll see where we're at. And if there continues to be interesting science to be done, I hope that we can continue this type of work into the future.
Yeah.
But I think equipping states and CDC and others who are serving like their public servants, um, that's also a goal of ours, because we don't want to just, we want to do translational work. We're teaching people how to make these measurements and how to interpret the data, and then they can take it from there.
Yeah. So just at the current point, like how long do you anticipate WastewaterSCAN continuing, either in its current form or evolving to more different research questions or something like that?
Yeah, like all science projects, we have like a time horizon of about a year. But depending on how things evolve, we might be able to go a little bit longer. So I can say with certainty we're going through, you know, at least through half of next summer, and we'll see where we're at then.
Thanks. Yeah, I think people will be glad to hear about that. Yeah, also to the points about like more different, looking at more different viruses. You mentioned H5N1 work. I'm curious if you could talk about that a bit more for people who might be unfamiliar with the research there. And also the challenges,
Yeah.
because I understand the animal dimensions of that virus are maybe scientifically interesting.
Yeah,
But
so
yeah.
H5N1 is avian influenza, which right now seems to only be causing like eye infections in people, which is great news. But it has recently this summer started infecting cattle, which was like a brand new thing. It was only really affecting birds, hence its name, avian influenza. And so we noticed this really weird thing late last spring is that in parts of Texas, which had been in the news, you know, having cattle infected with avian influenza, these super high influenza A concentrations in the wastewater. And we're like, what is going on? This is so weird because influenza season is over. And so we had an assay for H5, which is the marker in avian influenza, and we tested those samples and they were very positive for H5. And what we finally figured out is that plants in Texas were receiving raw milk uh, byproducts from like cheese processing plants or plants that were making cheese or cottage cheese, and their waste was going into the sanitary sewer, and that had markers of avian influenza. So this new use case for wastewater arose where we actually can track some viruses infecting animals if their animal waste ends up in the sewer system.
Ah.
So that's sort of the story, and we've been measuring H5 in wastewater since I think May or June. I've lost complete track of time as usual, and it's really exciting. The CDC is very excited about it. I think they're going to have their own map of the H5 detections using our data on their website soon, which is really cool to see that a federal agency is so excited about our data that they want to show it on their website. It's really great. So I think this new use case for wastewater that we can sort of look for viruses that are transmitted in animals and can potentially infect people is kind of cool, and we'll see what else we can do with that.
Yeah, I mean, I've talked to researchers, mostly to other countries, not so much in the US, who are looking specifically at sampling at like animal like farming areas or like slaughterhouses in the case of poultry. And so I feel like that will be another emerging area of research in understanding like what this data means when you're looking at different
Yeah, I
different kinds
think
of animal populations.
I'm an environmental engineer and environmental scientist, and I've always studied pathogens in the environment. So it's very cool to see that people are starting to get very interested in so-called environmental surveillance, which is sampling for pathogens outside the human body to understand their occurrence. And so like I find that very fascinating myself.
Yeah. Another question that came up in the Q&A that I thought was interesting is like looking more at different diseases. Like, are there other diseases that might have better data that link with transmission risk, and or like how do we continue to try to understand that link between the viral shedding in the wastewater and infected people? Like, is it maybe more clear with diseases like norovirus, where
Mm.
the symptoms are so like gastrointestinal symptoms? Or are there other diseases that WastewaterSCAN is looking at studying that might help to kind of understand those dynamics more?
Yeah, that's a really good question. I'm not sure I have a good answer for it. But I think it's a good like thought process. Like maybe SARS-CoV-2 is tricky because there's more asymptomatic infections. Is there a disease that has only severe illness, like for example, where there's really good clinical data that you can link up with the
Yeah.
wastewater? And I think the answer is yes, but I can't think off the top of my head of an example. Maybe like Mpox would be one because, or Ebola, for example. But yeah, that's a great idea.
Yeah. Yeah, I mean, I think this goes to another question that also I see in the Q&A, which is like that pairing of wastewater data and clinical data or like other kinds of health system data. So I'm curious like what kind of role WastewaterSCAN has or that like other researchers working on wastewater surveillance might have to kind of advocate for more like clinical testing, or I've talked to some researchers about things like wanting to do regular survey, like phone surveys, trying to figure out how many people actually have COVID right now in a given community, and like pairing that with wastewater data. I'm curious if that's something that like you all have capacity for, that's kind of on your radar.
Yeah, I mean, we haven't, it's not something that we've included in our wastewater scan like research plans necessarily, but as academics, like we recently got access to there's a research version of Epic, which some of you might be familiar with. It's like the medical records. When you go to the doctor, they fill in all this info. They have aggregated it and compiled it for researchers to access, and we just got access to that, and that's a great source that we're
Nice.
using to compare to wastewater. And I think doing like really careful studies where you're monitoring wastewater in a community and doing that kind of like surveys about health would be really great. And we've written like in papers in the discussion section, that is a great idea for future research. I think the trick is, putting together a nice proposal and getting funding for that from NIH or CDC. That's something that takes a lot of work. But
Yeah. And also like
would,
having the interdisciplinary team of like the
yes,
epidemiologist plus.
plus the environmental
Yeah.
scientist. Yes,
Yeah.
exactly.
Yeah.
And that's a challenge, always a challenge.
Yeah. I saw, hopefully this is a quick question. Someone asked if there are plans to measure polio virus.
Yeah, we're not, the answer is no, not yeah. No, we don't have any plans to measure polio right now, but it's something that we could do in the future. The next virus we're kind of looking at right now is Mpox clade 1, which is the virus that was just declared like a public health emergency in
Yeah.
Africa. So that's sort of the next thing that we're working on the research side.
Cool. And then one last one that I wanted to get in, because I think a lot of people ask this, is how, people watching this webinar or watching later can get involved with advocating for more wastewater testing, like in their community or just in general in the US.
Yeah, that's a great question. I know that people can always contact like their representative or their senator and and just advocate for it in that way. Talking to local politicians, I think, is a great thing to do. Um, I think I'm not an expert in this, and I think other people on our team are better experts. But I think even just like promoting this kind of information through social media, socializing it with people so that it becomes part of the daily discussion about like what's going on in the community. The more people that are valuing it and understanding it, the more likely it is to be future proofed. I think
Yeah.
so. Those are some suggestions.
Yeah. I know I can also offer from my reporting that one thing scientists like yourself have emphasized to me is like the wastewater treatment plants are also very important parts of
Mhm.
this work. And often they are like contributing samples kind of for free as like a extra part of their day-to-day jobs. And so, if you can find a way to like go directly to your local wastewater treatment plant or your like sewage authority, environmental, like local environmental authority, that's often a different branch of government than the health department. But they generally need to be involved to do this work. So that's another
Yeah.
kind of tip that I've gotten. Yeah, so I think we're at the top of the hour. We tried to get through a bunch of
Mhm.
questions, but I also copied some down that we can try to get to like later in writing. Is there any other like final thoughts you want to share for everybody?
No, just thank you so much for listening. And if you have questions, you can reach out to us at our website. We have an email, a director's email, which maybe Amanda can put in the chat in the last minute here. Or
Yeah.
you can Google me and send a note and we can get back to you. So thank you for your interest in this work. And thank you so much, Betsy, for organizing this webinar.
Yeah, thanks to everyone at the WastewaterSCAN team too for helping out with planning this.